So, what are we doing really? Ask a basic question, and we are tracking the filtrate, filtrate, okay, as a result of what? Ultrafiltration, where? In the Bowman's capsule, and we have got the filtrate, okay, and the filtrate is flowing along with the proximal convoluted tubule, we are still in cortex, okay, and then it will go into the, it will descend towards the medulla, okay, and then you will be in the loop of Henle descending limb, okay, and then you will take a U-turn, and then it will be an ascending limb, thin, still thin, and after little it will become thick, and then you will have a distal convoluted tubule, and just as you have the distal convoluted tubule, you talk to your own Bowman's capsule by a very specialized group of cells called as, very good, what do you call it as? Loudly, macula densa. What do you call it as? Macula densa. Okay, and then you continue as the distal convoluted tubule, you are still in the cortex, okay, and then you join the cortical collecting tubule which is in the cortex, and then go into the medullary collecting tubule, and then you are in the pelvis and that. So, what we are really doing is, you see, the each Bowman's capsule, each nephron has an enormous duties to perform. Like what? Almost look at every molecule, find out whether I need you or I do not need you, where do I need to take you back, how is my homeostasis in the blood, okay, if I keep you, am I disturbing my the homeostasis, how much, and in how much percentage each molecule or ion has to be there. Okay, so kidney has to, we have that elaborate system of tubules, the epithelial cells of the tubules, and those cells are continuously interacting with the filtrate as it passes. Okay, then you have to decide whether how much of water is there. Okay, you have taken 1 liter of water, ingested 1 liter of water, there is suddenly too much of water in your blood, the blood is getting diluted, its osmolality is falling, so the kidney has to make sure that no, I need sodium, potassium need to be conserved, particularly sodium and chloride need to be conserved, but the water needs to be excreted. Okay, so the kidney has to, and what we are really doing is, what are the mechanisms which the kidney, the cells along the tubules, what tubules which we have seen just now, how does those cells interact with the filtrate, and how do you deal with it is the question that we are asking. Now, I am here because to focus on a very interesting point. We are looking at the capability of the, so I do not have to explain this diagram to you. There are certain molecules which we do not want, body does not want. In particular, we take drugs, okay, we take drugs. The drugs we take them because there is a problem in our physiology, so we resort to pharmacology and take a drug, okay, so that our physiological processes are ameliorated, or in a sense homeostasis is restored, and the function of breaking down those molecules is mainly performed by the liver. It has huge number of enzymes. It can look at any molecule, find out where I can cut it so that it becomes small, so that I can send it to kidney and kidney can throw it out. If I can't do that, then let the molecule go into the bile, from the bile into the into the gallbladder, from the gallbladder into the duodenum, and then finally to the outside. So these are mainly the two options, okay, which by which the molecules can be thrown out of your system. Particularly, it has been found that some of the molecules like bile salts, oxalates, urates, and catecholamines, epinephrine, norepinephrine, okay, even they are readily secreted by the tubule into the nephron. Secreted, okay. Are those, where those molecules ultrafiltered? They could be filtered, okay, they are small, they could be, but those which are not filtered could be actively secreted. Now, this property of the tubule to actively secrete a certain range of molecule comes with a very interesting dimension. I will tell you why, it is really very interesting. The point is, even some of the molecules, particularly I will draw your attention to the molecules like penicillin and salicylates. So when I say salicylate, I mean aspirin. What do I mean? Aspirin and penicillin molecule, those molecules, if they happen to be in the kidney, the tubule is so efficient it will readily throw away everything from the tubule, okay, into the ultrafiltrate and throw it out of your body. Are you with me? What is the consequence of that? Can somebody help me to work it out? The consequence is that if you take a penicillin tablet now, the chances are that in 2 hours everything is thrown out. Are you with me? So, the biological half-life of penicillin and aspirin is so short in the blood because you have the problem there. So, does it mean that you should take penicillin tablet every 2 hours? Okay, that's a problem which the pharmacy people have to answer. Okay, and therefore we have a very elaborate branch of pharmacy which you call as biopharmaceutics, in which they find out as to how do we, what is the, you can look at the molecule, in what way you can change the molecule slightly so that its biological activity remains, but its biological half-life increases so that the patient can do with a tablet having every 8 hours. Are you with me? So, I have told you a very interesting dimension of this process of secretion of a large number of substances. Okay, very interesting. See, you would not believe till 1990, okay, 88, 90, okay, it is not too far off, pretty. How does water molecule go? How does water molecule go from this side of plasma membrane to that side of plasma membrane? And the general feeling in the science was that the water molecule is so small that it can cross the plasma membrane in spite of having charge, no charge, whatever, there is charge on water molecule, but it can still cross. Why? Because the molecule is too small, and if there is enough osmotic pressure difference across the membrane, the molecule can go. But that still did not explain that across some cells there was bulk flow of water, bulk flow, you understand what I mean by bulk flow? And one classical example of the bulk flow is RBC. Water just flows as if there is no membrane. How can water flow so easily across the plasma membrane of an RBC was a question. And the answer to that question was provided by whom? By whom? Okay, and what did he say? He discovered a system of protein molecule which sits in the plasma membrane of many cells. Okay, either it sits there or it can be trafficked. It can be trafficked so that it is there if I want, and if I do not want, I can remove it. How that happens, I will tell you a little later. Okay, and those molecules were called as what? Aquaporins. What do you call them as? Aquaporins. So, aquaporins, we have not known them for a very long time. Okay, Nobel Prize of Chemistry 2000, it is very recent, I mean 2003 is very, okay. So, what does he say? He says that this is a protein system that sits in the plasma membrane, and if the protein molecule is there, in that the water molecule can pass 1, 2, 3, 4, one at a time. It is not opening a big channel with thousands of molecules go. Hello? You get the point? So, look at that molecule, okay, look at that protein molecule through which the water molecules are going in what? In what you call a single file. What do you call them as? In a single file. Why should water go from one side to another side at all? What is there to drive the water? I have to give the answer 10 times. Osmotic. There is nothing else. There is no water pump there. Hello? There is no water pump there. Okay, it is osmosis. Okay, you will dilute more osmolality, less osmolality, the water will go. That is it. So, now let us see about why this is very important is because so far to my knowledge a total of 13 aquaporins have been cloned. Today if there are 2 more, I do not know. Okay. So, how many I am aware of? Good enough for our work. How many are there? 13. We also do not care about 13. For we put all them into one category, aquaporin is aquaporin. Okay. Now, this is a look, I want you to be smart enough to have a photographic memory and memorize this figure. Okay. Now, it is very easy, I will tell you why. I do not want you to bother about 1, 2, 3, 4 aquaporin. You see what he has given, you see aquaporin, one is what, say red. So, where is red dot, wherever there is red dot, what is author trying to convey? That is where aquaporin, I do not care, you do not care, okay. Okay, for us aquaporin is aquaporin, it is etic. Now, you ask very carefully, are aquaporins there in the Bowman's capsule? No. Are aquaporins there in the proximal convoluted tubule? Yes. Are the aquaporins there down into the loop of Henle? Yes. Now. Are aquaporins there by the time the loop of Henle takes a U-turn and as it starts ascending? Yes or no? No. Then you go into the thick part of the loop of Henle. Yes or no? No. And then you go into the proximal convoluted tubule. Yes or no? Yes or no? No. And then you go into the collecting duct. Yes or no? Yes. Yes. This has to remain with you, okay? This information, okay, has to remain with you. Without that you can't understand how it is possible for the kidney to make, if you have taken 1 liter of water, make dilute urine with osmolality of 50 milliosmoles, or if you are dehydrating, okay, you haven't had water to drink, then the same kidney is capable of making urine which is 1400 milliosmoles. Just look at the difference, 28 times difference. What, look at the capability of kidney, lowest as dilute as 50 milliosmoles, as strong as what, 1200 or 14 milliosmoles. Okay, you can't really understand why how kidney does it if you forget this diagram. Have I emphasized enough as to, you have to remember where the aquaporins are and where the aquaporins are not there. Good. Now, we are looking at, you see, if you remember, so far we have focused on what? You know, we have done everything what happens in the proximal convoluted tube in the previous lecture. We know how glucose is absorbed, we know about the GLUT, hello, we know about SGLT, hello, SGLT, okay, and then we also know that how the bicarbonates are absorbed, how creatinine is not absorbed. We have the background of proximal convoluted tubule, and now we go on, look at this diagram. The author has darkened this part, which is what, descending part of the loop of Henle, which is the thin segment, hello, thin segment, okay, all right. So that is what, this is an image from Guyton, and we are going to look at this. Number 1, this is the thin segment. What do you mean by the individual cells are thin? The individual cells are thin. By that you mean, and the the apical surface of the plasma membrane, it is not thrown into manifolds. So overall, the proteins that take part in active or primary or secondary transport, no, they aren't there really. No, they are not really there. Okay, mitochondria very few. Okay, all right, but proximal convoluted tube descending. Aquaporins, yes or no? Yes, aquaporins, yes or no? Yes, and therefore you can immediately understand what? Water can go out. Why will water go out? Water will go out only if outside osmolality is more. Hello? Otherwise water will not go. Okay, how that happens I will tell you eventually. But water, you fully understand the meaning of that arrow now. Why? For 2 reasons. There are abundance of what? Aquaporins, number 1, and outside the tubule there must be higher osmolality that can draw, okay, that can draw the water, draw the outward there. About 20% of the filtered water is reabsorbed in the loop of Henle. In this part, about 20% of the water is absorbed. Okay. Now we go to the ascending limb. Okay. In the ascending limb is this is the part of the tubule where minimum action is happening. Minimum action, not much. Why minimum action? First of all, it is thin limb. So are there any mitochondria? Many mitochondria? No. Okay. Is the inner tubule thrown into folds? No. Are there lots of enzymes and pumps? No. Okay. Are there aquaporins? Yes or no? No. Are there aquaporins? No. No. Is water freely flowing out? No. This difference between the descending and ascending limb has to be firmly ingrained in your mind. Okay. So as we are going up now, we are in the medulla. As we are going up, there is. But as you go from the thin limb into the thick limb of what? Loop of Henle, descending, thin, thick. That is the end of loop of Henle. Now I am done from the ascending thin. I am going to where? Thick limb ascending. Now thick limb ascending is a different. Cells are big. The cells are thrown into, the inner wall is thrown into what? Brush border. So a lot of enzymes are there, and abundantly equipped with all kinds of pumps and abundantly equipped with mitochondria. Lot of action is going to happen there. Now what action happens there is, there are lot of systems, symport and antiport or whatever, which are going to pump what out of the lumen of the lumen in the thick segment of the ascending limb. You can read for me there. Sodium, calcium, potassium, chloride, bicarbonate, they are all being pumped out. They are all being pumped out. Okay. So as a result of that, the osmolality is going out. Hello. All these ions, they will add to the osmolality. So there is huge amount of all those factors which will add to the osmolality are going out. Is the water following that action? No, water is not following that action. Water remains there. Water remains there. So in the thick segment, the water remains there. As a result of that, osmolality of the fluid as it is ascending in the thick ascending limb falls and falls and falls, and the whole thing becomes very dilute. Therefore, it is also called as diluting segment of the nephron. What did I say? Diluting segment of, why is diluting segment is diluting segment? You are absolutely correct, because it pumps out all the ions. So let us see what pumps out. Okay. So how does that happen? How does that happen? Here is the moment this image comes, the first thing you should look at, the moment that image comes, first thing you should look at, where is the lumen? Hello? Where is the interstitial side and where is the capillary? You are okay there? So look at that image, and this is the tubular lumen. So the filtrate is going, okay, now we are in the ascending limb, okay? Where are we? So I'll say that the fluid is going this way. Are you okay? Are you getting my imagery? The fluid is going that way, okay? And the fluid will come in contact with hundreds of cells. One of them is drawn here, and on the on the apical side or with the filtrate, there is a system. And what is that system? That system is, I already told you, we have done this system once. Can you tell me the abbreviation of that system? NK. Very good. What do you call it as? NK system. Does NK system itself use ATP? No, it does not use ATP. It is a secondary pump. Then what is the primary pump? Primary pump is here on the basal side. You can see that. Okay, that's on the, and one thumb rule which I have given to you. What was the thumb rule I given to you? No. The pump is always on the basal side. The pump is on the basal side. So check whether the rule I stated applies here or it does not apply here. Can you see the ATP there? Okay. So why is this pump why is this secondary transport working? Because this pump keeps on working. It throws the with every cycle of utilization of single ATP, you are going to pump out 3 ions of sodium and take in how many ions of potassium? 2 ions, and as a result of that we have the concentration of sodium ion falls. As a result of that, sodium from outside is drawn in, and it goes, because the sodium ions just can't go on their own. So you have this system of proteins which we call as NKCC. Are you okay so far? Now this is a protein. This is a big protein here. I'll show you the image there. There's a big protein, huge protein. It goes up and down. Several number of times it goes in the plasma membrane. Outside means on the apical surface where that protein is coming out. That protein actually comes in contact with the filtrate. And it will come in if it comes across with there is a binding site on the protein where sodium can combine. Another site, okay, where potassium can combine. Another site where CC. There must be 2 sites. Hello. NKCC, there must be 2 sites, and it is only when all these seats are occupied, then the protein undergoes a configurational change, okay, and then all the four ions NKCC, are you with me? They are taken from outside to inside. Who is the driving force? Sodium. Why sodium? Because there is a huge amount of sodium here, very less sodium here. So the power comes from the sodium, the difference in the concentration of sodium and the charge. Because of that, the protein molecule undergoes a configurational change, and all those things are delivered to the outside. So what am I showing you? Now listen to this. This is becoming more interesting. What am I drawing here? There is a protein here. Huge protein molecule here. This is just a symbol. There is a huge protein molecule there. Are you okay? And there is a site where sodium can combine. There is a site where potassium can combine. There are 2 sites where chloride can combine. There is a drug, okay, which is very commonly given for the treatment of high blood pressure, okay, and they are called as loop diuretics. What do you call them as? And there somewhat the philosophy is that those drugs increase the amount of urine formation, okay. So you throw a lot of more fluid from your body, and when you throw more fluid from the body, there could be relief on the blood pressure. Are you with me? Hello. This is one of the strategy, not a great strategy, but we have to do with what we have, okay. Such molecules are called as what? Loop diuretics. What do you call them as? Loop diuretics. So when a patient of high blood pressure, doctor prescribes him loop diuretics, some of the agents which are available in pharmacy are given there. Can you read the name of the first drug for me? Say that loudly. Furosemide. What do you call it as? Now that furosemide combines with the protein molecule at the site where chloride is supposed to bind. Hello, hello. So this is the molecule. On this molecule, there are two sites where Cl and Cl are supposed to combine. On those sites are now being occupied by what? There is a competition between the molecules, chloride and furosemide. Are you with me? But the binding of furosemide in that competition, furosemide wins. So does the chloride have an opportunity to bind with the space? No. And if and the beauty or whatever of the pump is, even if one of them is not there, the pump won't work. The pump won't work. So you have literally inhibited the pump. Okay. So because you have inhibited the pump, the chloride doesn't go, then the potassium doesn't go, and the sodium also doesn't go. When the sodium doesn't go, the sodium remains in the tubule. When sodium remains in the tubule, proportionately water remains in the tubule. And when the person passes the urine, more water goes away. Therefore, we use the word diuretic. It is a diuretic. What do you call it as? Diuretic. And this particular class of drugs which have their mode of action by specifically inhibiting the protein. What is that protein? NKCC. Within the nephron, where is this protein located? Where is this protein located? In the thick segment of the loop of Henle. Thick segment on the loop of Henle. Is it on the apical side or on the basal side? Apical side. Very good. Good. All right.
So, does this work for the NKCC in the drugs?
In the? Which one? Furosemide. It works wherever this pump is there, it will work. Yeah, it works there also. So we are looking at the same molecule. We have the again look for the ATP, look for the basal side, look at the apical side. I given this because I want you to read this description. It tells you how okay. We are looking at the NKCC molecule. Just can you tell me how many times the protein you have to appreciate the protein molecule. How many times it's going up and down? 12 times. Okay, and it allows what? Okay, one of this, and two of this. We'll move on now. So what have we done just now? We have tried to understand how the fluid in the thick segment of the ascending limb of the loop of Henle is being treated being handled by. Okay. Now if you go early, so now so this was the thick segment. Now I am going to the now we are in the cortex, and I'll call this as, what will I call it as? Early. We are in the cortex. Cortex of the kidney. What do you call it as? Early distal tubule. Now early distal tubule is interesting because that's where the macula densa will begin. That's where macula densa is. We are familiar, but we are not talking about macula densa now. Okay. Then forms a part of juxta that we have already done. Is avidly reabsorbs most of the ions, including sodium, potassium and chloride, but can you read there? Is virtually impermeable. Why? There are no, you are right, there are no aquaporins. Therefore, this is still a part of diluting segment. Why is the diluting part of this segment so important? I have told you the answer 10 minutes back. I just want the answer back from you. Because, yes, because if you have ingested a lot of water, 1 liter of water over a period of 10 minutes, we can do that. Okay. If you, but within a matter of another 15 to 30 minutes, no, okay, let's see. What is the osmolality of human blood? 300 milliosmoles. A little wrong, but good enough for government work. It's about 285, 290, okay, but 300 is okay, good, 300 milliosmoles. So that is osmolality of the blood, that is the osmolality of the plasma, that is the osmolality of extracellular fluid, that is the osmolality of the intracellular fluid, 300, good, everywhere 300, easy to remember, good. Now you have to ingested 1 liter of water, okay. As a result of that, from 300, within a matter of water has gone to the stomach, absorbed, and then within about 15 minutes you will find that the osmolality has fallen from 300 to 290, okay, or maybe 285 or maybe 280. Okay. Now we have osmosensors. Now, how osmosensors work? It's in the next lecture. Osmosensors. Okay, there are neurons in the brain which will monitor and homeostatically tell the brain that it's a little too much of water. Okay, and then So the kidney will say, okay, you need to form dilute urine and you have to throw out. Had it not been for the diluting segment, it would not be possible for the kidney to form a dilute urine. Are you with me? And why do you want to form a dilute urine? Because if you do not form a dilute urine, along with the urine we will throw a lot of sodium. You do not want to do that, okay? Okay, you see, you have taken water, how much of sodium was there in that water? Very little. Osmolality of the water that you drink may be 10 milliosmoles, it is so dilute. If you have taken 1 liter of that water, okay, you are diluting the whole system. So your kidney sees, amazing organ, it sees to it that you get rid of water, but it also makes sure that you do not lose salts. For doing that, you have the diluting segment, where it is water, relatively less salt, and if you keep it like that, you throw it out. So what really is thrown out of the body is what? Extremely dilute urine, okay. But the beauty is we have only 2 kidneys, and the same organs have to function on one hand if you have taken too much of water, on another hand if you have not taken water, okay. Then how does your kidney deal with the other extreme, okay, is something. My time is done, we will talk about it in the next class.